Hello friends, welcome to my channel. This is a video on the reproduction of angiosperms. Now in this process we come to pollination, which is transfer of pollen grains from anther to the stigma of the flower. The formation of pollen grain or male gamete is a step-by-step process which is completed in 4 steps. In step 1 you can see the development of microsporangia, which are the cells of hypodermal region. They become differentiated as archisporial cells. The archisporial cells get further divided along the periphery to form outer parietal layer and inner sporogenous tissue. You can see the figure over here. The outer parietal layer gives rise to wall of sporangium along with epidermis. The innermost wall of this is called tapetum. In the figure you can see this is the tapetum, this rounded structure, this bluish colored structure, these are called tapetum, which nourishes the developing pollen. The endothecium layer help in dehiscence of anther to release the spores. Endothecium means this one, this is the layer, this pink. This is endothecium. Now in step 2, there is formation of microspore or pollen grain. The sporogenous layer divides multiple times to form diploid pollen mother cells, which undergoes meiosis to form 4 haploid pollen grains. These are called microspore tetrad. The structure of pollen grain has the outer layer called exine, which is a thick cuticularized layer, contains resistant fatty substance called sporopollenin, which can withstand acids and alkalis. It is a highly resistant substance. The inner layer, it is called intine, it is smooth and made up of cellulose. Here you can see the figure of pollen grain. This is the outer exine, which is rough surface consisting of sporopollenin. This is the intine, this black one. It is a smooth layer. It consists of cytoplasm, and this is the nucleus. There are germ pores. These parts are called germ pores. These parts are not covered by exine, free from exine. Now in the step number 3, there is a development of male gametophyte, which takes place within the anther lobes. When half of the germination is completed, the anther dehisces. The germinating pollen grains are shed. After the dehiscence, the germinating pollen grains are shed from there. After that, microspore undergoes mitosis to form 2 unequal cells. The larger one is known as vegetative cell and the smaller one is known as germinative or generative cell. The vegetative cell forms pollen tube after landing on the stigma, means after pollination. The generative cell or germinative cell forms 2 male gametes after pollination. Now in the step number 4, that is the development part after pollination, which is the formation of pollen tube. Here you can see this is the pollen grain which has formed this long tube like structure. This is called pollen tube, which actually comes out from the germ pore part after landing on the stigma. This is the stigma. After landing on the stigma, the pollen tube comes out. The pollen tube contains 2 parts. The one is tube nucleus, which comes just at the peripheral part of pollen tube, and there are male gametes. So this is the development of microspore. Now coming to step-by-step formation of female gamete. The first step is development of ovule. The process is known as ovule is known as megasporangium. It takes place within the ovary and remains attached with ovarian wall through a structure called placenta. The central part of the ovule is called nucellus. This is the structure that is formed. This is nucellar tissue. It is mound-like structure which develops to form, the nucellus grows bigger and gets covered by two walls known as integuments. These are attached to the placenta by a central stalk called funicle, and the nucellus is covered by two integuments, two layered. Next is formation of megaspore. It comes from megaspore mother cell. The sporogenous cell acts as megaspore mother cell MMC, which undergoes meiosis to form 4 haploid megaspores. Out of these 4, 3 of them get degenerated to provide more nourishment to the developing ovule, and 1 become functional to form the embryo sac within the ovule. This megaspore this is the first cell of female gametophyte. As you can see the formation, step by step formation. This is one nucleated which undergoes mitosis division called and forming 2 nucleated stage. This 2 becomes 4 nucleated stage, and finally it becomes 8 nucleated stage, in which we have this structure. This is embryo sac. It contains 3 antipodal cells. Antipodal cells on the chelazal end. This end is called chelazal end. In the middle there is a 2 polar nuclei. This is a fusion product which is diploid structure. Here also there are 3 cells to the micropylar end. The middle one is called egg cell, and it is flanked by 2 haploid cells called synergids. So this is the development of female gametophyte. So thank you guys. Please like and subscribe my channel, and don't forget to leave your comments. Thank you.